First, thank you very much for an invitation. It's a pleasure and an honor to me to be here to share my research at University of Florida and to share what I do in my whole life. I'm going to talk a little bit about where I came from, how I got here. But first I want to ask something important. How many of you are commercial beekeepers? Commercial beekeepers. 1, 2, 3, 4. Okay. Oh. Okay. Because I am Dr. Humberto Boncristiani. I'm the applied honeybee researcher at University of Florida. My research program is dedicated to commercial beekeepers. I help the big guys. I work whatever they need, whatever research projects we can come up with, I help them get richer. Let's put it this way. So today, I want to talk about something that I saw a lot of misunderstandings when I travel all over the state of Florida to work with commercial beekeepers. And it's a subject that people have a lot of doubts and questions. And I think it's a good opportunity for you guys to learn more about viruses. That's one of the biggest puzzles on biology, what viruses do and how they cause diseases. And that's what I'm trying to do here today. In a very slow pace, the same way I talk with my father, which is also a beekeeper. So let's go slow. I want to invite you guys to go to the world of small things, the smallest things ever. All right? Okay, a little bit about me. I'm Brazilian. I grew up as my father is a commercial beekeeper in Brazil, small commercial beekeeper in Brazil, and I grew up around the Africanized bees. So the Africanized bees were the normal to me for many years. So as you can probably imagine, I got stung a lot. But we learn fast. So,
they sting you in the head also?
Oh, I hope, yeah, sometimes, yeah. Yeah, they sting everywhere. You know that. But kid, I never liked them very much, especially because when you're a kid, you don't like to get stung for many years and got in trouble for hiding, all kind of stuff like that. And I avoid the bees for a long time. Then, okay, no more bees. I go to college. I did biology. And then there is one job for me there is about bees. I say, oh, crap, here we go again with bees. I got the job. Then no more job. I moved to my master in my PhD in a medical school to work with viruses. I do vaccines for babies. No more bees. Great. Work with the viruses. I was doing vaccines. I was a happy person. Then in 2006, the bees start to die around the world, and apparently it was a virus. At that time, they thought it was a virus called IAPV, Israeli Acute Paralysis Virus. And the virus that I was working on my PhD was a cousin of the one of the bees. So they're looking around, the USDA were looking for a guy that have the background of beekeeping and also virologist. And apparently I was the only person on earth. So USDA brought me in from Brazil in 2007, and since then I'm doing research on bees. So I give up. I'm a bee guy, I'm a bee person, that's it. And that's what I'm doing since then. This is some photos of the medical school where I got my PhD, working with viruses in Brazil and in São Paulo City. And this is the virus that I was working. They are called enterovirus, they're the family of picornavirus. They cause all kinds of nasty diseases in humans. Meningitis, encephalitis, paralytic diseases, carditis, and all this stuff. And I don't know if you saw, there is a word, paralysis. And if you associate with honeybee viruses, you see a lot of viruses that have Israeli acute paralysis virus. And almost all the viruses have some paralysis on that. There is a great number of honeybee viruses that cause paralysis. And they are very similar And this is the same family that cause polio disease. So start to understand the association. The bees that got paralysis, human got paralysis from the same family. So they share similar biological trends. And I'm going to try to go over this very smoothly without the same way I try to do with my dad, and he doesn't shoot me. So yeah, I already talked about the transition. I was doing my vaccines, and then the bees start to die. There is this paper on Science publication talking about IAPV that was very correlated with the cases of colony collapse disorder in 2006. And then the USDA were looking for a postdoc with virology background, and that's the way I come in United States. So yeah, here we go. That's the family tradition. Bees everywhere, but we need the big suits. Trust me, we need those. So and since I'm here, I worked 4 years at USDA. Then I moved to Greensboro. I got a grant from National Academy of Science. I work at the University of Maryland with Dr. Dennis Van Ingelsdorp. So I've been around the country traveling a lot, working with bees. I work for a private company too, trying to develop nutritional systems for honeybees. So I've been around a lot. And something that comes all the time is that why I'm doing this, keep doing this, is I'm keeping a promise. I hope you guys have to do that, but when you leave your country and you need to talk with your dad and your dad come to you, you ask you, what are you doing that? And my answer was, I'm going to help the bees. And then he looked at me and said, okay, help the bees. And that's what I'm trying to do here. I have a lot of history of being a troublemaker, but that's what I'm trying to do. And that being said, finally I can talk about this freely without problems. There is a little conflict with the university. When the university hired me, I was doing a parallel project, a personal project that there was a possible conflict, and I might need your help. I don't know if you guys know me from the YouTube channel called InsideTheHive.tv. It's a project of mine that I want to keep going, making videos to help beekeepers about bee biology, the last research papers. I don't know where I'm going with this yet, and I don't have too much time. But now the university gives me free, the dean signed the paper, I can do it. So great. So whatever you guys need for videos, go to the website, sign the email list, tell me what you need. I'll make a video about it. Okay? That's my way to spread the word. We have the technology, so I can reach more people. So that's what I'm doing. Please show me some love there. I appreciate that. Okay. Now, another thing that I like to, in all of my talks around the state of Florida, I like to thank you all for the contribution, because with the effort of all the beekeepers, the state, and everybody involved, you guys helped to build one of the biggest facilities to study honeybees in the world. It's an amazing place. I don't know if you guys have the opportunity to visit, but Florida, you guys, thank you. Beautiful place. Thank you very much. And I'm honored to be part of this team.
We built that. Yes, we built that.
Yeah, you guys built that. Yeah, please. Top facilities in the world, and I have been in lots of them, everywhere, Asia, Middle East. I've been everywhere working bees, and this is high level, top level. All right. What is this? Yeah, I know, it's sad, but we need to talk about the sad things so we can get rid of them. This is a bee with the fawn wing symptoms, with a lot of varroa all over her. Poor baby. And what I'm trying to do here today is explain how we got that. How a virus can get in and cause this mess and all these variations that we see. The same virus can cause many different kinds of symptoms and different timings and different locations. What I'm going to try to do here today, very simple. I'm going to try to be very simple, but I need your help. imagining things with me. If you think I'm very fast, please let me know. I stop, I come back. Because at the end of the day, I want to pick your brain. Maybe we can get some ideas here so you guys can help me. How can we get rid of this? If you know how the biology work, how the pathogenesis work, meaning how the virus caused the disease, maybe we can come up with some ideas here, and then we can discuss at the end of this presentation. Do we have a deal? All right, fantastic. So as we know, the bees are suffering from many different things right now. There are a lot of different variables that we didn't have before. I talk with commercial beekeepers and they always remember, oh, the old times, we just go, leave the bees there, go fishing, come back and get the things from nature. It's very easy, life is easy. 60 years ago, something like that. But things have changed. There is a lot of things going on. The world is changing, the nature is changing, there are a lot of different variables that we didn't have to deal before. And you're going to understand that all those variables combined cause the variability of symptoms and everything that we see, and especially viruses are master to take advantage of these things. And I hope with this presentation I can give you a hint of understanding of how that works. So what I'm trying to show here is very simple. Life is very diversified, and we like to separate things because we like to put things that is equal together. So we have a protobacteria over there, archaea, animal cells, and they are different. They are different from each other. They evolve in a different path. So they're supposed to be different, like elephants are different from us and things like that. In the microbiology world, they're different. We have bacteria that are different from fungi that are different from animal cells, and so on and so forth. And viruses, what is virus? That's the thing, it's one of the biggest puzzles. Because what is that? Because this is not something that is, some people even say that they're not alive. So the way we define something in a very simple way, that a virus today is a biological entity consisting of a genome of a DNA or RNA. Don't be scared with these molecule names. I'm going to tell some of those, but you can forget about it later. Just consider that stuff, okay? Let's call this bad names stuff. DNA or RNA that encapsulate with protein to protect them from the environment. That can only replicate inside of a living cell. So outside the cell is a stone, is something. It's not alive, do not have any metabolism like a bacteria. Bacteria can go and grow with sugar. Virus cannot do that. They do not collect things from outside, put inside for themselves. They don't eat anything. They're just a molecule covered with the protection system. Simple like that. And they're waiting in the environment until something happens and they reach the right cell. But we're going to get there. And they're very diversified. Viruses have different sizes, different shapes, and they do different things. One have protein plus lipids, other have all kinds of shapes and forms. I'm not going into the details here, but the important is the shape and the form and the structure and the material is very important for their biological agenda. What they need to do is find the cell and replicate. That's their biological agenda. And the way to find the cell is about the capsid, the protein. Whatever is covering and protecting them is what I call the address. It's going to be there in nature. It's going to until it got there. I'm not going too fast, too slow. Okay, let me know. And when we are talking about viruses, people have this misconception that they know it's small, but they don't have a good idea of how small it is. Let's give this as an example. You have an orange here. like it's a human liver cell. And you imagine this all over. So we're talking one cell, and I'm trying to give you a perspective here. And then in green here, we have a bacteria, which is the last thing that we can see in a microscope. It's very small. Bacteria is very small, and it's this size, and the human cell is that size. Then we have the viruses that are even smaller. We cannot see in a normal microscope. We need a special microscope to see viruses. And you have all kinds of sizes and shapes. And guess what honeybee viruses are? They are this one, the smallest one. Picorna viruses. Pico, it means small RNA. The genome of them are RNA viruses, is small RNA viruses. They are the smallest virus on the planet, one of the smallest in the planet. And to have an idea about size, one thing that I like to imagine the Palm Beach County. This is a human cell. Soccer stadium. This is the bacteria. The virus at the ball. Got the idea? We're talking about something small, very small. And this is a structure of one of these honeybee viruses. They're very simple. They consist of 4 proteins, 3 outside and 1 inside associated with the genome, which is a RNA. And they form this icosahedron. I don't know if that's the way to say that, but they protect this genome, which is a RNA, that have the information for them to replicate. When they got in the cell, this thing is released, and then they dominate that cell for them. They use all the resources of the cell to produce more viruses. So, and something that I like to stress here is, this is very powerful. The structure of icosahedron in nature is one of the most strongest structure you can possibly have. Diamonds have things like that. And there is a reason for that. They are not bacteria that can run from things, or they need to be stable with rain, with whatever happened. When a bird eats a bee, need to pass through the digestion system of the bird. We don't know how that works yet. We're just starting this research. We don't know where these viruses go before they come back to the bees. So we're still learning from that. So something that I like to say is that the capsid is the address and the key to enter in the cell. Is a specific cell. We're going to get there soon. But remember that. I'm going to come back to that later. The address and the key to enter in the cell. This is a little illustration that I did when I published in this review on the respiratory viruses in an encyclopedia of microbiology. That was old, but it's still okay. Let's. In yellow here we have a cell, and these are the virus. Can you see there is? I will jump. Don't laugh at me. Right here. When there's touching, the virus, every virus needs a specific receptor in the surrounding of the cell, in the envelope of the cell, which is the door to get in. So we don't know about them yet. We don't have the resources to do the research that is needed to be done to find out which receptors are that, because that will be a fantastic tool. If you know the receptor, we can come up with some chemical that stop that. But we're still very, very in the beginning of honeybee virology world. So we don't have the tools yet. We're working on that. I'm also working on that. There are hope that we can find those and try to block them. We're working on that. So, but very basically, so they got in. Once they got in, that capsid opened, and the genome go straight to the cytoplasm. And then the cell recognize that as a self, recognize of their own material, and start to produce the molecules, the proteins of the virus instead of the protein of the cell. So start to consume the energy of the cell to produce things that are not going to help the cell in any way. And very simple, I just want to, the take-home message is, from this family, if you do association with other families that we know those numbers pretty well, one virus got in, 100,000 get out in 7 hours. Do the math. One cell, one get in, 100,000 get out, 7 hours. Imagine how many viruses you can make with one bee body. We're talking about a lot of viruses. So when we're talking about viruses, it's always good to assume they are everywhere, especially this family, picornaviruses, one of the most successful virus family in the world, like common cold. I shake your hand? No? And then you have common cold. They are here. You touch it.
That was pretty bold.
Yeah. Oh. So they are very tough. They can survive in nature pretty well. We have common cold. We have a lot of viruses that have the same mechanism of transmission. They need, you touch the bees. We can assume things like that. So for the human family, that's the way transmission happens with picornavirus sometimes. But with bees, it's a social. They interact. They share food. So you can imagine that one is contaminated. It's very likely that everybody has it. So just keep that in mind, that because they're social and they share food, it's very, I would assume in my beekeeping operation, if you send samples out to somebody and get a positive, assume that you have everything. But the good news is, viruses are tough. But we're talking about the most successful society in natural history, honeybees. They are here for 30 million years. We are here for what? I don't know, 10,000 years. We are starting to understand how can we stay together and happy. They are here dealing with crap for 30 million years or something like that. Can you imagine? They have defenses. They develop strong defenses. When I go to the lab, one of the things that I like to say, I'm not going to tell about this because somebody's going to shoot me. All right. Okay. Let's continue here and then I go back there. I want to talk about misconception about viruses. Viruses do not have metabolism. Please don't ever do that again. Come to me and say, "Oh, are the viruses going to eat my honey?" No, there is no such thing. No such thing. Viruses do not eat anything. They enter in the cell, they replicate. They don't eat. They don't have metabolism. Okay? No more of that. I don't want to. It's common. It's unbelievable. Yes, sir.
If a virus can be prevented from entering a cell, does it live? Does it die? I mean, what happens?
It wasn't alive before. How could it die?
No, I think what he's trying to say that if he do not enter, if he's going to stay there until he got an opportunity to enter, if there is some mechanism that can get rid of them, I would say yes. If they're inside the bees have ways to always monitor and protect themselves and clean themselves. And when they don't have and they get infected, the bees have something that is one of the most amazing things. Bees are able to recognize they're sick and they fly away to die alone to protect the hive. It's one of the most beautiful things. And I think it's some of the lessons also that we need to learn from this society, that we need to be together in this, because if we're not together on that, we are probably going to be in trouble. Yes, sir.
There, in the last picture, there was the word mushroom.
Mushroom?
I'm hoping that they
Difficult
kill
to
these
treat mushrooms.
mushrooms.
Where's a mushroom?
Over there on the right. Where you
Okay. Okay. Yeah. Difficult to treat. Yes, mushrooms. Yeah, there is this mushroom going on. I don't know much about it. But everybody asks about that. I don't know much about it. I know there are some results. That was very enthusiastic. I saw the patent, so people are serious about that. So one of the ways to see if somebody is serious, if they're paying to protect that idea. So when you have a new product, go see if you have a patent. Because if you don't have a patent, the people don't believe about that product. So yeah, I saw the patents. There is some hope coming up. But personally, I never tested myself, so I don't have much to say about it. All right. No shooting. Yeah, no shooting. No metabolism. Okay. When I started studying honeybee viruses, they were around 14, something like that. Today we're reaching 35, almost 40. So I'm not going to all of them. I'm just going to tell you that there is a bunch of them. You probably heard some of these names. Acute bee paralysis virus. Remember, paralysis. Israeli acute paralysis virus, paralysis. Kashmir bee virus, black queen cell virus, sac brood virus. There is a bunch of them, and they're separated based on the organization of the genome. Don't shoot me. I know, but it's important because I want to show something. How a molecule that can be so simple and so small compared with our genome, of the genome of the insect, can cause so much damage and at the same time be so successful in nature is one of the beauty of these things. And I want to share the beauty of viruses with you. Don't shoot me. All right. So the structure of the RNA, the genome, is very simple. We have a bunch of proteins, a bunch of genes that code for proteins responsible for the replication of the virus. And then you have a bunch of proteins, genes that code for proteins to form the capsid. 1, 2, 3, 4 proteins to form that capsid, and a bunch of other proteins to do what? Dominate the cell and replicate the genome. Simple like that. And very small. Very small. One that is in, the whole metabolism of the cell
Becomes it.
hijack. Yeah, that's the word. So, however, some members of honeybee viruses are a little bit different, and they're the opposite. Instead of the replication proteins to be this side, they are in this side. And because of the organization of the genome, we put them in another family, for example, Iflaviridae, which is the formal wing virus, and varroa destructor 1, and the ones that cause these diseases that I showed the picture. Remember, my goal here is to go with the biology a little bit, so you understand a bee getting the virus, entering the cell. I want you to imagine all of this and come with me so I can guide you how the wings got twisted, or how the bees are smaller, or we're going to get there. All right. I don't know. This is here again. Here you go. And another thing that is important, and the way to explain that is very simple. Our genome is DNA. It's supposed to be very stable. When you replicate our cells, we have mechanisms to check if the replication is corrected. So we don't have much mutations going to one to another. We have a very small amount that is kind of good. It's important to have some mutations to not be completely equal. But when you have a genome that is RNA, you don't have the proofreading. So every time a virus replicates, is different from the other one. And remember the one that I said? One get in, 100,000 get out. I'm talking about 100,000 that are a little different from each other. And that's the power of the genome of them. Because that small amount of genome can compartmentalize a little information, but when they replicate, that can become huge information. Because now we're talking about a cloud of information that small viruses that are a little different with the capsid, maybe they can get in another cell. Maybe when they replicate in the other cell, they replicate again and become different, and they can get another tissue, another insect, another organism, and keep going. RNA viruses are powerful because of this process of mutation. No proofreading. But some guys, very smart in NIH, discover something that they use for HIV, for example, that we need to have a threshold. Because if you mutate too much, those proteins do not have the function anymore. They're completely different.
Yeah, so is there about how successful are those mutations? Is it like,
The environment is going to select. That's the power. They produce a lot of different things, and wherever they are, at least one of them is going to be successful. And that's the strategy, the biology strategy behind RNA virus. They produce a lot of variability, and then at least one or a couple of them are able to survive in their environment. That's why they're powerful. That's why they're everywhere. They're everywhere in the world. And sea, land, animals, insects, they're everywhere. This kind of family, it's everywhere. And what I want to show here is basically what I said. If you see this ball and then the blue and then the green, each one of these is one replication cycle. One get in, 100,000 get out, and from these 100,000, and then you can see that they can create those diversification. That's the main what I want to show here. I already tell the end of the story, so I know. A lot of diversification, and that's very powerful for them. Moving on. And what I want to say is just 1, 2, 3, 4 replications cycle. You have a huge variation already inside the bee.
And you can use a high-powered microscope to see that?
Yeah, you can see them, but they look very alike. What we're able to do, we compare the genome. We're able to sequence those genomes and compare, and we see the differences. But yes, ma'am.
When they multiply like that, are they not only multiplying but adapting to their environment to try to survive?
The environment is going to select the ones that are able to survive there.
So they're altering really to survive. They're not just altering to reproduce.
They are altering because what they do, I don't see an intention, but that's how nature works. They produce a lot of different things, and nature kill all of them, and whatever survives, survive, and keep going. That's the way it works. Yes, sir?
The whole reason they make so many different varieties, so that way one of them can be compatible to
Wherever they are.
Those are all random changes.
Random changes, yes, random changes. We try to control that and see, but it is random. The data is there. We never know where in the genome that's going to happen. Okay, so let's try to go one step further and try to answer this question. So why now? Because before, the bees were fine. And they probably have viruses anyway. As I said, they're here for 30 million years. So I'm for sure, the same way, the bees that survive this whole thing are the bees that have mechanisms to survive. They have protection. So the bees, when they feed something and have viruses, they have different pHs, they have different enzymes in the digestive system, they have mechanisms inside the cell to fight these viruses. But what have changed? Well, what changed is that along all this evolution, when the bees were just feeding viruses and they have protection against, 50, 70 years ago, something, when Varroa arrived, 100 years?
87.
Something, yeah, here in the United States, yeah. Now, all those defenses that were selected is gone because Varroa inject directly in the fat bodies and the hemolymph. So all the defenses the bees had before is just useless. That's why Varroa is a problem. It's not mostly because they feed on the fat bodies and less energy, yes. There is all of this, and they consume energy of the bees, yes. Just like us, mosquito took out for us, but our problem with mosquitoes is not the mosquitoes. It's when we get some dengue virus and when we get stuff that we start to bleed. That's the problem here, and it's something that you guys need to understand. Control Varroa right now is the only way to control those viruses, but we can talk about that. So when we inject them, you need In a normal infection, when I give viruses for them, I need 10 to 8, that's, I don't know, 100 million virus particles to make them sick by feeding. I just need 100 particles to make them sick by injection. 100 or 1,000 particles, that's, I don't even know. 100,000 times less virus. 100,000 times less virus to do the same damage just because you change the position where they get in. And all the defenses are gone. And that's what the problem is right now. Yep, this is the guy. I'm sure you guys see this a lot around here. I'm not going to get into the details, what you should do with...
Make sure you look for them.
Well, yeah, when you see them, it's too late. You normally should do your counting when you don't see them to make sure, because when you see it, that means that's too late. There are too many already. You got the message? Viruses, varroa, why varroa, the problem? Okay. Now I want you guys to think with me, because I know you guys know bee biology, because everybody's beekeepers here, and I want to try to exercise to imagine how we got that. How we got that. So, okay, we have varroa that have the formal wing virus that feed on the bees and get in the bees, and then what happened? Where do you think it should go? We have bees, different life stages. We have what I'm trying to say is, when varroa is in the cell feeding on bees, he starts to feed in different life stages sometimes. So the injection in the larvae, but even in the larvae, one instar different from the other, and there is some variation of timing. Timing of infection, there is some variation. Variation number 1 is timing of the first infection. Variation number 2 is the amount of virus they'll get in. Varroa have different amount of virus sometimes. Sometimes they inject one virus, sometimes they inject more than that. So we have another variable, the injection. So the timing, the injection, how many varroa we have there. The third one is sometimes there is description. I'm not very confident about that, but there are some articles that say that the virus replicate in the varroa first and then come back. If that's true, this is a third variable, because when you got replicating in a different tissue, you remember they're going to come and become something completely different. And then they're going to be injecting something in the bees that's completely different. And the cycle continues. So for you to see something like that is only happening in a very specific case. Because a lot of people come to me about, look at here, my bees. There is 1 or 2 bees with that, and they assume the others do not have. That's the only virus that you can see symptom. And it's always good to assume that everybody is infected. But you see symptoms, you need to get this in a very specific case where the virus are able to reach in that specific development stage to cause that. If the virus is not there at that specific time, that's not going to happen. Remember, in the metamorphosis of the bees, the tissues change, the cells change, the receptors change. So there is windows of opportunities for the virus get in the cells and come back. It's not all tissues that the virus can get in. It's only specific tissues with a specific receptor in specific time. So all these combinations cause, when you see this, is because the virus were there in the right amount, in the right time, in the right time of the bee development to get access to that. But when you don't see it, doesn't mean they're not infecting other tissues inside and doing other kind of damage that we cannot see. But you're only going to see when your bee is going, boom.
They can affect maybe the digestive system.
Digestive system.
Or the honey sac.
Or the energy, you have smaller bees. And then with time, all this progression. And again, I forgot to mention, we're talking about the virus mutability here, but they're master because we have that cloud of information. Remember that I said they produce a lot of different things? Pesticides. If you have a pesticide and the bees are consuming, dealing with the pesticides to get rid of things, that expose different things, that change. And everything that change in the tissue or in a bee, it's going to have one of those viruses that adapt to that situation. So when you have UV light, when you have a different chemical, we never know when in that cloud of information you're going to have one that's going to boom and just kill everything. So the way the bees deal with that is very kind of similar. Bees reproduce, the queen mate with lots of drones, so they have a lot of genetic diversity too. So it's not all the bees. The chances are that not all the bees gonna be sensitive for that. So the ones, again, the virus is gonna be able to infect them and kill a bunch of them, and they're gonna replace with the ones that are strong. So we always have this balance, the selection. I like to advocate always for better queens. I don't like chemicals, I don't like things, but in case of varroa, and commercial guys need these things, they need to put food in their table. So I don't want to go into the details of who is right, who is wrong on these things, but I'm a believer of we need better queens. We need better bees. We need bees adapted to the situation that we have in the world today. Otherwise, you'll always be consumed with products or things that are going to make our life hard. But I'm not saying it's easy, because the biggest problem in the world is human beings. This is a collective effort. If you one messed up, done. So that's why I'm not very optimistic about the possibility. Who wrote it? Yeah, yes, sir.
A recent studies by Dr. Thomas Seller, The Life of Bees.
Yes.
He does a lot of studying of wild honeybees.
Yes.
And it seems that because taking into consideration the decline of the colonies under management, okay, versus wild bees, wild bees seem to go ahead and
they are selected.
themselves, and they actually live with the varroa virus, the varroa attached to their bodies, and go about their businesses like they didn't exist.
Yep. There are a pressure for natural beekeeping. They call it Darwinian beekeeping. There is pros and cons, but he's right. If you let the bees do what they do best, we're going to have better bees.
Did it for 30 years ago.
Yeah. They're always doing that. That's the way they survive for so many years. I want to have understanding why you have so many diversity of symptoms sometimes. And it's the same virus. For example, this virus that I worked with when I was in a medical school, depending where you inject them, you got a different disease. If you give by mouth, you get encephalitis. If you get by injection, you get something else. Depends what kind of tissues they reach first and do the diversification again. So I want you guys to understand that and go home and think about that for a second, because this is happening all the time. We have plant viruses with similar, we have all kinds of living things that are going down and up. And in nature, it's always about the balance. We need balance in this world. We just need more balance and more for me. We need balance. And I think that's what I have for you guys today. Thank you very much. Now we're here for questions.
Are you going to take some questions?
Yes, questions. If somebody has questions, then I will try my best. Yes, Jesse.
I was wondering about an antiviral for the mites.
Antivirals for the mites.
If the mites don't carry a virus, then they don't affect the bees.
Yes. When I said that I'm not...
They're going to have to be small.
Yeah.
That needle's gonna be real small.
The problem is the delivery method. We need something that go in and do not cause any harm to the bees, and only the bees can consume that. And the Monsanto is working on that, and that kind of thing. But again, I think the bees have something that is much more powerful, which is the mechanism they develop along the years. If you want to be a slave of a new product, up to you. I don't defend that things. I think we can. Management of bees is something that I learned even more working with commercial guys. I can guarantee you, if you know bees and you know how to keep them, the bees are going to be fine. Yes?
Once you see one of the bees is mutated, is the whole colony affected?
The bees?
Yeah. Once one of them is mutated, is the whole colony affected?
Are you talking about the virus or the bees?
Right.
That could be. Depend of the genetic of the bees that we're dealing that, and the genetic of their virus. If there is a bad combination, bees die, but also the virus die in the process. So there is always this recycling circle that the bees keep doing, and we need to help them to do it better, that in a way that do not kill the colony, because that's the mechanism of defense of them. They have some room for losses. Yes, sir.
Are there different seasons where the viruses spike or drop? Kind of like when the snowbirds come to Florida and bring all the colds with them? It seems like flu and things like that.
Yes.
Back from California. Yes, there you go.
There is some theories behind that. Autumn going to winter, when you have more varroa, and the bees start to have a longer living period, so it's more time for the virus to be cooking there, and the bees cannot fly away because they're overwintering. That's a time where the bad things happen. When the bees cannot do what they do best. The sick bees go away. It's just the cooking of viruses. You're cooking viruses in the winter. And that's why you need to be very strategic when you're controlling your varroa, because you need to enter in the winter with very low amount of varroa. Otherwise, you're going to be cooking whatever was there.
Bees who weren't grown on varroa.
Sorry?
So very low levels of varroa, but also bees that were grown.
Yes, yes. Yes. If you keep selecting bees that naturally...
time period from when you treat to going into winter, you got to make sure there's a good hatching.
Yes, you need a good amount of bees, but you also need a low amount of varroa. Yes, sir.
What mechanisms do the bees have as far as immunity to dealing with these viruses?
One is the one that I told you, they fly away, altruistic mechanism. We have an RNAi system, RNA interference. Inside the cell are able to recognize double-strand RNA. Okay, you ask, don't shoot me. Inside the cell, I show you there is RNA, the genome is one molecule. But to produce that molecule, the cell needs to produce a template, which is the negative strand. So we use the template to produce a lot of the genome of the virus. When you have that hybrid, the double-strand RNA, an RNA that's complementary to another one, cells are able to recognize that, and they have a system to these things. So this is one mechanism they have specifically against viruses. Yes, sir.
The strength of the hive to a weaker hive, I think Roland goes to the weaker hive, not to the good hive that's got a lot of bees.
I think varroa goes where they have food. Brood, you can be weak, strong. I don't think you can differentiate that. They need brood. That's what they need.
But varroa don't walk
They fly with other bees,
Yeah, so like a robbing or a...
robbing, yes.
A weaker hive might be more susceptible because they got less bees to.
It's not published, but I think I can talk about that a little bit. We did a little experiment in the lab with the observation hives. We paint the bees with different colors and put in observation hives, in 12 of them. And in about a month, we have 40% of different. Everybody was 40% equal. There was a mix of everybody. So we're talking about when you have a yard, they are kind of one thing.
Drifting.
Yeah, drifting is just a lot.
Professor,
Yes.
how far apart were the entrances? Approximately.
That was this,
2 or 3 feet?
but I work a lot with a good friend of mine, Randy Oliver. When we did the experiment, we could see different, very high numbers with almost 1 kilometer far away.
Still the same results?
Still lower, but still they
Are you talking about robbing or drifting?
Drifting, no robbing, drifting. Yes, yeah, drifting. That's important. So yeah, they like to share.
Well, they could
things.
go to the same flowers and stuff like that.
Yeah,
Yeah, but they end up in different hives when they come back.
Yeah.
Drifting.
Yeah.
Did everybody understand what he said? He said at the bee lab, if you've never been there before, they have a whole wall of observation
Yeah.
hives. And the observation hives all have entrances going out to the outside. Well, all of those entrances are about 3 feet apart. They marked bees inside of each hive, and then a month later, like they marked them all a different color. So entrance number 1 was blue, green, red, blah, 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 blah. Every hive had their own color, right? Or some sort of similar
Yes.
kind of marking. A month later, they go back, and they can see that every hive has lots of colors in it. This isn't the red hive anymore. This is like, they're all rainbows now.
How many days exactly did you check?
I think it was 30 days. I didn't
How many days did you check this?
He said
for a whole month. I think it was
How long the bees were living? If you say in a month, you were still seeing bees.
I didn't follow the experiment. It's not my experiment. That's the result that people told me. I don't know the details, but I can get it for you later on.
Because if they fly, means they can live after this only 2-3 weeks. So how come they are still alive in another?
I think
They mark the young bees, they mark the whole hive.
we follow them for 30 days, but the drifting occurred very fast in the first week or so. We could see drifting very fast. I can give you more details later. I'm gonna put the note here.
Now, the mice are existing hundreds and hundreds of years.
True.
The guy was Jacobson who first described it, correct?
Correct. In Asia, yes.
And those bees got adopted and they were surviving because they were leaving the hives. They were swarming a lot, leaving the hive,
Yeah,
leaving the nest.
that's one of the hygienic behavior is a big component on how to control bees naturally. For example, in Brazil, the Africanized, we don't have the same problems that have here.
They were going away from the mice, leaving the hive, establishing a new hive. This is how they survived.
Yes.
Now, did they have the viruses back then?
Good question. Maybe.
Did they have viruses? Because if they had viruses, that wouldn't save them for changing the nest.
But you're
It
forgetting
just
one
was,
thing.
hold on please.
Okay.
It was just only the mice that were affecting them. So probably it was not viruses back then. Otherwise they take the viruses with them.
In other words, there might be variables.
Chicken egg. Who came first? That's what maybe we're talking about here. There was an occasion somewhere, because that happens too. It's not it's the same variation that we're talking about. I told you guys that the bees are strong when they got by feeding, they have defense, but they got sick too. And what might have happened is in one of these occasions when one of those that got viruses by feeding got sick and spread all over her body, and you have the mite in that position, was the first time where the mite get the virus. And then he start to spread. Do you understand when this starting point happened? In some occasion, 100 years ago or something like that, viruses were there and one bee got sick. And that sick, when I get sick, I said systemically. The virus were everywhere, in the hemolymph and the fat bodies. And in a random event, the mite got that bee and get viruses. And from that point beyond, things start to get worse. He could start to spread.
Special virus that
We don't know.
attacking the bees. This virus was attacking the bees. The bees have to be attacked in order to get the mites infected with the virus.
Yes, that's.
And then the virus, when he chews on the larvae, he infects the larvae. This is where the problem is. It's not when the bee is already adult.
Oh yeah, no.
The virus affects the larvae.
Yes.
It doesn't affect the big bees.
It does. It does. Just measure them. Get a bunch of them and measure and weight them. They're smaller. When another symptom is the formal wing virus, the bees, because you can see, is very. You need them. You need the machines to do that. They're smaller. They're lighter.
But that comes when they hatch out. They're smaller and lighter, correct?
Yeah, could be because they have less material to build up.
Is the older bee when that varroa is on the older bee that's already there doesn't have any effect on that older bee when they're hitchhiking or when they're sucking on.
Well, yeah, they're still feeding on it.
Yeah, it's still feeding on it.
But it's not causing any virus at that point.
What he's saying is that phenotypically, the disease is expressing itself in a different way.
That's true. But I think you mentioned do not cause problems with adult bees.
Yeah, the virus.
The virus. What is that photo then?
No, I'm just asking.
Yeah, no, I mean, I'm trying to...
Get the viruses when they are in the wound, or when they are alive, they also can get infected by virus.
There is ingestion. But for that, the bees are kind of strong against that because that's the way they got viruses along the evolution, before mites came in.
And some of the bees come out with a short abdomen. That
Yep.
was the description
Yep.
30 years ago when nobody knew about viruses anything. Because the mites are sucking out everything, so she is born not developed.
I agree with you, sir.
So it was done by mites at the beginning, and then later, somehow, somebody mixed in this virus.
Well, you had a host shift with those mites.
I'm enjoying that because every single example that you guys are talking about happened. So it's a lot of things that happened at the same time.
But what he's talking about is that years ago, when the mites were only Varroa jacobsoni, and they were on Apis cerana, and then those eastern honeybees and western honeybees occupied the same area, and a mite.
Yeah, it was a jump. Was a jump?
From the eastern to the western honeybee. Now all of a sudden you got a
different bee.
type of mite on
And
a
a different
type of bee. The whole game.
It's a party. It's a party. It's a party.
But the Varroa causes multiple problems. It's a vector for disease, but it also causes its own problems for the bee.
Yes.
The only problem is not the virus transmission. It's the Varroa is also its
Well,
own
how could you
damage.
separate that out? If every Varroa is carrying viruses, how do you separate out?
Well, Varroa affect the bees in many different levels. That's true. When they're consuming the energy supply, so the bee's going to have less energy supplies. Fat bodies are responsible for, is a very unique tissue that responsible for many different things: detoxification against pesticides, defense against microorganisms. So when you consume that tissue and you have less of this tissue, you have less power to deal with all of these things. So this is another thing that varroa can cause problems too.
Is it, what is affecting more? Let's say we have 5 mites in this cell.
Okay.
And then we have young bees that are teething. And because the young bees didn't hatch out 100% the way God made them, so they don't produce the right jelly,
Yes.
the quality of
Yes.
So this is causing more actual damage to the larvae than the mite itself inside.
In a social, yes, you're correct. They can't work the same way a normal one would work. They kind of handicapped somehow.
That's why sometimes we make a split with a beautiful queen, but the brood is not healthy.
Something is there.
So who is at fault? The young bees that cannot raise a new generation to replace themselves, or the amount of mice? So maybe we should watch very careful and not to produce such bees that are not capable of feeding properly the larvae.
Who did that? I think somebody presented on that at ABS about switching the queens. Like you have a beautiful hive, beautiful brood.
Yes. Marla Spivak, yes. They did an experiment, if I remember correctly, just to demonstrate that what people were thinking was a weaker queen wasn't the case. You just put the weaker queen in another hive, and apparently everything come back to normal. It was something in that hive that was affecting that queen for her to not produce the same amount of eggs. I don't know the details of the study, but that's the beauty of bees. The bees connected with the queen, and the queen communicate with that. Virus get in the middle of that, mess up with this communication. Pesticides come in, open opportunity for a new virus come in. It's a mess. We have too many things going on. It's a mess. It is a mess. Welcome to my world.
Way back in the back.
Yes, sir.
The study that you did with drifting, when you had painted the bees.
Yes.
What time of year did you do that? And then,
I think it was a couple months ago, 4 months ago.
was it here in Florida?
Yes, in the new bee lab.
Well, North Florida or South Florida, because we're different 4 months ago. We're still in the 80s down here.
Okay.
Why do you ask that question?
Yes.
I'm just curious to know if it was a dark, if it was a honey flow, what time of year, winter, summer, it was in this state.
Yeah, we know that happened. We are we need to expand the experiments now to see if it happens in every time of the year, in every condition.
You can do from what side the rain was coming. If they are working over there, they see the rain, and they are coming back. But on their way will be another hive, like you say, a mile away.
They stop
They
there.
will all jump into this hive.
Yeah, they're smart.
Out from the rain.
That's true. That's true. They found the conditions. If the conditions are not okay, they find the first hole they got in.
Conservation of energy.
Yeah.
Show up with the present.
Yes, sir.
I got food, let me in.
I can't hear you. How queen? Yes. Yeah, they can get sick with viruses, you mean? Yes. There are cases that we saw sometimes queens also with the formal wing. So queens, black queen cell virus is a virus that was, the name of the virus is because the queen larvae got black. So that's where the name of the virus came from. So yes, queens got, yep.
Chalk brood is from the queen?
Yeah, the chalk brood is a fungi. That's yes, sir.
You gotta replace the queen to fix it.
Say it again.
4 different colonies that all started.
That was 12 or 16.
So were they all working as the same, like one colony all together? Like can they each go from colony to colony without no problem?
When we saw every colony have all the colors.
Wow.
A colony will let another bee in if it has food. If it goes empty-handed, it isn't going to let her in.
Well, I can tell you too, that from experience, when I started my business, I would expand my business by starting a new bee yard. And I would take splits from one bee yard and move and start my new bee yard with it. And I would take all queenless splits, or I would think they would all be queenless. I'd take them to the new bee yard and I would then put my queen cells in. Every once in a while, there'd be a queen in there, and the next day you'd come back and you'd be like, I think that one has a queen in it, because it's covered in bees. And the bees want to be on the winning team. A lot of times, if the hive is queenless, bees will drift to a queen right hive. That happens.
Absolutely true. That's true. They don't want to die and...
To a queen that has more
That happened to me. The younger, better
A
queen.
hive that has a queen with more pheromones will attract more bees than an old failing queen.
That's correct too. So many things going on. Yes, ma'am.
So if we kind of like left the bees alone, like you said, they were like millions of years ago. If we had just left them alone, us as humans,
Okay.
would we have that problem now? Because it's kind of like, did we get in the way?
Well, it's...
We would be pretty hungry right now because we wouldn't have bees pollinating our food over here.
It's hard to predict.
But like they were stronger when we left them alone, right? And they were fighting off the viruses, you said?
It seems complicated, but it's very hard to predict. But what I can tell you what's going to happen. Some's going to die, some's going to stay there, and if everybody died, they got extinct. That's simple like that. So whatever happened in the environment, whatever happened in this planet, we never know what's going to happen. That could happen with us. Tomorrow, we got a virus, a pandemic virus, and get rid of all of us in a month. Well, Ebola is not the biggest problem. Ebola kills a lot, but do not kill too many. Like in 1918, the flu, the Spanish flu, 1/3 of the population was gone. Yes, sir.
Did I understand correctly that your personal opinion is to change the queen to a more, to a better queen?
No.
Resistant to,
Now you want me to lose my job. Okay. Personal opinion. Okay. I like the idea of to select bees, to have better queens. We have ways to do that, selecting queens. That's why we breed. That's why we can leave it alone, but then we're gonna need to get rid of a lot of beekeepers because the bee population is gonna go down a lot until we get other bees that are gonna take place. But we can select queens. I can bring another subject that somebody's going to shoot me, which is genetically modified things, but we don't need to go that. We know how to do these things to speed the process. But I'm enjoying the idea of getting better bees. I think I can put this in a way that can keep my job. All right.
So Tom Seeley's book that was just mentioned a little while ago.
There are 15 of them.
Okay, the one that just came out a week ago.
Oh, okay, the new one.
The newest one.
Okay, I didn't read that one.
A couple of days ago. Okay. He says that years ago, he was working up in New York, and the feral bees ended up getting varroa, and initially, most of the hives ended up getting wiped out. But there were a few survivor hives that then went and swarmed and repopulated the forest that he was working in. But there was a big bottleneck of genetics at that time. And now all of the queens in that forest are descended from many fewer queens than they had been previously. Much genetic diversity in the feral colonies that have survived this varroa.
That's one of the downsides when I have a big drop. But again, bees are tough. I trust.
It's a good thing.
It is a good thing.
We had him here as a speaker, and he began his lecture by saying, how do you end up with more mosquitoes? Try to kill them. And you're right. You're exactly right when you say he's a good speaker. We've had him here.
Yes, ma'am.
So,
sorry, I came in a little bit late, so I hope you already haven't gone over this. But you're saying that the varroa mites are bringing in the diseases, and if we treat for varroa mites, the disease is still in the hive.
If you treat for varroa, you get rid of the varroa, and then you decrease the chance of varroa to cause more damage, transmit more viruses, or consume energy resources from the bees.
The virus
So
will
then are you suggesting
work
that we
its
should
way
be
out.
more actively treating?
I'm not suggesting anything.
Humberto,
Yes.
when you say what you were just talking there, because after if you kill the varroa, the viruses are still there. Aren't they digesting it as opposed to the injection method of delivery? And that should make it easier for them to deal with?
You're talking about varroa or the bee?
Well, she's talking about if you kill all the varroa, but the viruses are still in there. But if they're in there, they'd be consuming them.
When you say in there,
in
where?
the hive, in the colony.
Yeah, okay.
Say you got a colony, and you've treated for varroa, you've got your count down to 1 or not even 1. But they had the virus from the varroa being in there previously.
Yes.
So the bees technically still have viruses in them.
Yes.
But over time, after 25 days, 45 days, those bees are dying out and they're cycling out. And so that
New
virus
ones.
is going to cycle out.
Well, then we're gonna enter in another subject.
Maintain.
The viral load.
Yeah, but I want to touch base with this. I don't want to, maybe don't have time for that, but then I did this. I got hive tools that was 2 years in an old bee lab in Beltsville. And I get all the wax from that, and I decide, let's check if there is viruses here. That was there. The former wing virus were alive for 2 years in your hive tool. So you also pass things around. You do not inject any bees, but you pass yourself. The same way we do we transmit viruses. That's why you go to a hospital, there is all these procedures to, everybody have different clothes, and because we know that from our personal experience that we are able to transmit things too. The same in our apiary. So my suggestion here, every time you go from one colony to another, if you can burn your thing when you're a smoker, that helps. So yeah.
Flame your hive tool.
Yes.
What temperature kills virus?
If you boil the majority of the virus that we know today, you can kill them. The standards procedure from medical device like bleach 10% kill a lot of viruses. We are Florida State Beekeeping Association is working with us. They asked us if we could do some research based to understand if bleach, and this is cheap materials, could be used to sterilize whole frames. So we're studying that now, and so far I can bleach. I dip half of a frame in a bleach, pure bleach, and put straight in one colony to see if the bee is going to reject that. They didn't reject at all. The queen lay eggs normally in that part of the frame. So far, apparently it's fine. But now we're trying to prove if it kills everything. We know that it's able to kill bacteria, most of the bacteria that we have. The problem is to demonstrate that for viruses. So that's what I'm working on right now. I think I lost the original question. Please.
So you're saying treat the hive tool, burn the hive tool.
Yes.
And you're saying that, so it's straight bleach or bleach water solution?
Bleach 10% solution.
Okay, and freezing?
Freezing, that's the way we keep them alive. Yeah. In the laboratory, we get them and we freeze them. That's the only thing that some nosema are sensitive to freezing, but you're not going to freeze your colony.
No spores or bacteria.
So what you're saying is I should dump my whole hive in bleach?
Yeah.
Bees and everything?
No. Only for
Virus free.
various live free. What I'm saying is that might help for old combs, if you can spray them or clean them, you can use it again and do not propagate the diseases they have there. No, you don't want to kill your stuff. Yeah. Yes, sir.
I'm curious, going back to breaking the mite, different varieties of honeybees, some of them have a 1 day shorter cycle of gestation.
Yes.
How much work is being done on breeding queens that lay eggs that will mature in a day or two less than the current life cycle?
Well, if you go to the South Florida, you have the Africanizers that they're much faster on that. Yeah. That's one of the success of them is this ability to produce and to keep the engine running faster and produce more bees and swarm, and so there is no time for things to happen. But there is consequences for that too. Yeah. Yes.
When you didn't specify, when you said you dumped the frame in the bleach solution, did the frame have foundation and comb on it?
Yes, the wax and everything there, just to reuse. Yeah.
It was just an old frame that was
old frame, yeah. The ones that you keep to reuse again, dead out. Yes, we don't know what killed the hive, but you want to reuse the equipment, and you don't know if you're going to spread. We are working on that. So again, I'm not suggesting anything. Good. Yet. But when we get the answers, Mary is going to make a blog, and we're going to start to propagate that. And if they don't do, I make a video about it. All right. Yes.
So going along that same question,
yes.
I typically will take a couple of my frames that I'm not going to get all the wax off, and I'm not going to take all the honey out. I'll freeze it in case of. So you're saying that I could very well be freezing the viruses?
Yes, and you're keeping them alive forever.
You're not hurting the virus by freezing it.
You're doing it for hive beetle larvae?
Yeah, that works.
You're doing it for stuff you can see.
But I want to reuse those frames. That's okay.
But you're not doing it to get rid of the viruses. You're using it to get rid of any bacteria.
Small hive beetles and some other organisms that are sensitive to freezing, to cold, which is not the case of any virus.
Wax moth.
Yeah.
I just freeze mine to preserve them so I can use them again where they're not destroyed by wax moth.
Yes, wax moth. That's the way to preserve them. Yes, sir.
Excuse the question, but...
So is it about suggestions too?
5.5%.
Say it again, sorry.
In the supermarket, you get 5.5% chlorine. So that means 10% would be 1-to-1, like approximately, that'd be 10%?
No, you need 90 milliliters. Get yourself a milliliter cup, 90 milliliters of water, 10 milliliters of bleach.
Yeah, okay. Yeah, he mentioned something important. When you buy bleach, the one that you get, Clorox, I'm considering whatever that solution is, 100%.
Oh, okay, all right.
From that solution, you dilute 10 times, and it's still viable to kill things.
If you buy chlorine from a pool store, which is 20 bohm, I believe, it's different. It's a lot different.
There are different kind of Clorox. Yeah.
Yeah, but if you just dip a comb into a water solution, how do you get penetration into all of the cells?
That's my question. You must have to spritz it.
We sprayed. Yeah, that's the.
So it gets into all.
When I said I did, that was the first thing I did, and I got in that problem, and then I decided to spray.
Okay.
And I spray with 100%, not with 10%, with the full load, and right after that, I put in a hive to see what happens.
You didn't rinse it off?
Nope. I wanna.
They didn't care about it?
They didn't kill the hive. The queens were laying now.
And how much time?
The queens were laying, but they didn't burn their tongues.
I need to work with worst case scenarios, because when you publish something, there is always someone that's gonna do something that's the worst. So, yes, sir.
When you sanitize commercial dishes and things, you put them in a soap water put them in a rinse water, and then you put them in a bleach water and you let them dry, air dry. You don't wipe the bleach off.
Are you talking about restaurant, right?
Yes.
Okay, yeah.
Or a sanitizer solution.
You don't wipe the bleach off. It
Yeah.
doesn't hurt anything. It's going to dry it in the air.
Bees love chlorine.
Chlorine,
Every chlorinated pool, the bees just love it.
chlorine will evaporate and break down anyway over time.
Sorry, yeah. Yes, ma'am. Sorry.
Are you planning on doing research as to the queen when she does lay eggs in these frames that have bleach on them?
We follow that. We follow those bees after she laid to see if...
No, you're misunderstanding me. Drop in a frame into bleach.
Yes.
And you're doing a experiment with it.
Yes.
Are you planning on, in that experiment, looking at the queen and the cells that she lays in on that frame, the variables between a weak hive and a strong hive, the variables between the varroa treatment you're putting on?
Yes. Yeah, that's true. That's true. That's true. I don't have answers for that. We need to expand perhaps the experiment because I follow to see if the hive will... It was a very fast experiment to see if they lay eggs. The brood was okay. We followed the adults. Everybody was fine. So then we were satisfied at that point. Then we go to the lab to test against microorganisms. But you're right. We never know what time of the annual cycle of the bees that something can interfere on something. You always need to keep our mind open. Yes, sir.
Is the commercial hive and the wild hive going through the same things right now? Are they experiencing the same diseases?
Wild hives, that's a good thing. What is that? Yeah. Especially in Florida. Well, yeah, the ones that go to the bushes here, probably some that swarm from some commercial operation. Is that really wild? I don't think so.
You can't swing a cat on Beach County without getting a commercial.
Yes. So it's very easy for us to see a hive in a tree and think that they're different, but they might be only a hive that just get out from Sierra's bees, for example. Yes, ma'am.
Are any of those viruses or negative ramifications transferred into the honey? We scrape it and we spray it, it's not needed, it's not anything.
You find viruses in honey.
But it's not transmissible to humans.
For humans, no. Oh, we will know very easily that. Yeah.
We'll know when it happens.
Yeah.
We've all lost our way.
Yeah. All right. Thank you.